Hello dear students. Today I am going to teach you a chapter number 4. The name of the chapter is a Transpiration from credit number 1st from subject Plant Physiology. So the student, in which chapter today's lecture we covered the topics mechanism of closing and opening of stomata, steward hypothesis, active potassium transport mechanism, opening of stomata during the daytime, closing of stomata in absence of light. So the student, firstly I am dealing with mechanism of closing and opening of stomata. Students, in the last lecture we already focused on the structure of stomata. The stomata are nothing but the small minute opening presence on the upper and lower surface of the leaf, and these stomata, it consisting of the pair of guard cell along with the subsidiary cell. The process of stomatal transpiration are largely regulated by opening and closing of stomata. So the opening and closing of the stomata is called the stomatal movement. The stomatal transpiration occur through the stomata when stomata are open, and guard cell plays major role in opening and the closing of stomata. So the guard cell, these are the main cells within the stomata, and these guard cell, it is responsible for the opening and closing of stomata. The exact pathway of transpiration and the mechanism of opening and closing of stomata takes place as below. The first point, examination of opening and the closing of stomata under microscope reveal that the opening of stomata have a turgid guard cell. Those stomata are open, the guard cell, the nature of guard cell, it become turgid, while the closed stomata have a flaccid guard cell. So the size of the pore, stomatal pore, it is a depend on the degree of turgidity of the guard cell. When the guard cell are turgid, the pore is open, but when the guard cell become flaccid, the pore is closed. So the student, you always keep in your mind these things. So in order to understand the mechanism of opening and closing of stomata, one has to look at the submicroscopic specialized organization of the wall of guard cell. So the student, in a last lecture, I am already focused this point. The guard cells become kidney shape in a dicot stomata, and these guard cells, a differential thickening pattern, result in a very thick inner wall of the guard cell, while the outer wall is thin dorsal wall and a somewhat thickened ventral wall or pore wall is there. So the student, in a grasses, the dumbbell shaped guard cell is there, and this function like a beam with a inflatable ends. So the guard cell plays a very key role in opening and closing of the stomata. So the student, how the stomata get open and how it get closed. During the daytime, when there is sunlight is there, so the starch in the guard cell, it is converted into the sugar, means it is converted into the glucose. So in the presence of light, the guard cell, the product of photosynthesis, there is a starch is there, accumulation of starch in a guard cell, and this guard cell starch, it is converted into the sugar. This conversion takes place in the presence of organic phosphate, and during this the enzyme, the name of the enzyme is a phosphorylase enzyme, acts as a catalyst. Means this starch, it is converted into the sugar in presence of the enzyme phosphorylase. This reaction is carried out. Glucose being soluble in water. When starch, it is converted into the glucose, then glucose, it is soluble in water, and due to that it's increased the osmotic pressure of the guard cell. So the guard cell, after increasing the osmotic pressure, the guard cell take up the water from the neighboring cell. That neighboring cells are called the subsidiary cell, and due to the diffusion of the water in the guard cell, there is a continuous diffusion of the water in the guard cell. The guard cell become turgid. Outer wall of guard cell being thin and elastic, it get stretched or it get pulled outward, while the inner wall, it is a non-elastic, and the thick guard cell become a concave. So as a result, the two guard cells move apart from each other, and the stomata open widely during the daytime. So this turgid pressure or turgidity, it helps to pull out the elastic thin outer wall, and while the inner wall, it is a non-elastic, but then this guard cell become a concave shape, and due to this guard shell, they are move apart from each other, and the stomata get open during the daytime. When the stomata get open, the water vapor from intercellular cells is lost to the outer atmosphere. So this is the process during the daytime happen. The stomata, it get open. Then under the condition of darkness, that means the student in absence of light or during the night time, sugar of the guard cell is converted into the starch. There is a opposite reaction is there. In a daytime, starch is converted into the sugar, but at a night time or in a dark condition, the sugar is converted into the starch. Here the enzyme is a work, that enzyme is a phosphorylase. So that reverse reaction occurred with the help of enzyme. That enzyme, see here phosphorylase enzyme is there. This phosphorylase enzyme plays an important role, but there is a conversion. There is a the phosphorylase enzyme in presence of light, and see this conversion is also happen with the help of phosphorylase enzyme. So the starch, it is insoluble in water, and it is a osmotically inactive substance. Therefore the osmotic pressure of the guard cell does not increase, okay, and it undergoes the depressed or downward. As a result, there is a water diffusion from the guard cell into the neighboring cell. So this interconversion, the glucose, it is converted into the starch. Then starch, it is inactive osmotically, and that's why here whatever water present within the guard cell, that water comes into the neighboring subsidiary cell, and due to this the turgor pressure of the guard cell become decreases. As a result, the guard cell become flaccid, means whatever water present within the guard cell, that water moves or that water goes into the neighboring subsidiary cell, and this guard cell become a flaccid. Due to the flaccidity of the guard cell, stomata is a partially closed or it is a completely closed. Stomata close, and that's why there is a no transpiration. The stomatal opening become a closed. So the student, this is happen at the night time or in absence of dark condition. So the next I am focusing here, the steward hypothesis. That steward hypothesis, it is also called as starch sugar interconversion. So the stewards in 1964 proposed the modified theory. He shows that glucose 1 phosphate is osmotically inactive. And unless this glucose 1 phosphate is converted into the glucose, then no appreciable change in a osmotic relation can occur. So glucose 1 phosphate, it is a osmotically inactive. When this glucose 1 phosphate it is converted into the glucose, then there is a osmotic relation can occur, or there is a turgidity occur. He also shows that there is a metabolic energy in the form of ATP. That means the adenosine triphosphate is necessary for closure of the stomata. And see here, oxygen also required for the respiration to produce the continuous ATP supply. So the student see that the glucose 1 phosphate does not cause any change in the osmotic pressure of the guard cell, because the inorganic phosphate and glucose 1 phosphate, it is a equally osmotically active according to him. Here glucose 1 phosphate, see here the starch plus phosphate, with the help of phosphorylase enzyme, it is converted into the glucose 1 phosphate. And this glucose 1 phosphate should be further converted into the glucose 6 phosphate. And see here the glucose 6 phosphate, with the help of enzyme phosphatase, it is converted into glucose plus phosphate. See, glucose 1 phosphate should be further converted into the glucose, and there is a formation of inorganic phosphate during the daytime. So the metabolic energies are required in the form of ATP would be required for the closing of stomata, and this energy comes from the respiration. See, the guard cell carries the enzyme. The name of the enzyme is a phosphorylase enzyme, and this phosphorylase enzyme, then there is a phosphoglucose mutase enzyme is there. Then there is a presence of phosphatases and phosphorylases enzyme in the guard cell. These enzymes help in a opening and closing of the stomata. See, at a higher pH, the pH 7, the opening of stomata will be takes place in a following manner. See here, there is a the photosynthesis is there. Okay, then the result of the photosynthesis, there is a decrease in a CO2 concentration in a mesophyll cell of the leaf. Then due to the decrease in a concentration of the CO2, then increase in a pH of the guard cell. Then hydrolysis, see the when the pH get increases, it is come into the 7, then the hydrolysis of the starch into the sugar by the enzyme. See, the starch, it is converted into the sugar in the presence of enzyme. Then increase the osmotic pressure of the guard cell, and there is endo osmosis is there. Water of the subsidiary cells come into the guard cell, then increase the turgor pressure of the guard cell, and stomata gets open during the daytime. So this is regarding to the stomatal opening process. So the student, the stomata close. See, the glucose plus ATP with the help of enzyme hexokinase enzyme. Okay, this glucose plus ATP, there is a product is formed glucose plus glucose 1 phosphate. So the student, this glucose 1 phosphate, it is in the presence of enzyme phosphorylase, it is converted into the starch plus phosphate. And you know the starch, it is a osmotically inactive. The closing of stomata will be brought about by the conversion of this glucose 1 phosphate with the help of the ATP in the presence of enzyme hexokinase and the oxygen, and it is converted into the starch in the guard cell. So here, see here, this glucose, it is converted into the glucose 1 phosphate, and then this glucose 1 phosphate with the help of enzyme phosphorylase, it is converted into the starch into the inorganic phosphate at the low pH become 5. So this is regarding to the scheme of the Stewart hypothesis to explain opening and closing of stomata. So see here the starch, it is osmotically inactive. So the whatever water present within the guard cell, okay, there is a hexosmosis is there. That water it is goes into the neighboring cell, and the guard cell become a flaccid and the stomata get closed. So this is regarding to the opening and closing of the stomata according to the stable hypothesis. So the student, see the stable, see here the mechanism of opening and closing of stomata. See the student, see in a dark condition there is no photosynthesis is there, increase in a CO2 concentration within the mesophyll cell, then decrease the pH it become 5, then in that less pH acidic pH, conversion of sugar into the starch takes place and decrease the cell sap concentration, osmotic pressure undergoes decrease, hexosmosis of water is there, then decrease the turgor pressure and while the stomata closed. So the student in a next here in a presence of light, photosynthesis process is carried out. Due to the photosynthesis, here the carbon dioxide it is used as a raw material for photosynthesis, and the decrease the concentration of photosynthesis in a mesophyll cell while increase the pH, it is a near about the 7 pH, and here the conversion of starch into the sugar. So the student increase the cell sap concentration due to the uh, glucose, and here endosmosis is there. Due to the endosmosis, the turgor pressure increases in the guard cell and the guard cell become fully turgid and the pull out the wall of the guard cell and the stomata become open. So see this is also the cyclic condition I am showing here. See this is a day condition student and this is the night condition. So see this is the day condition. Okay, the starch it is converted into the glucose 1 phosphate. It is become converted into glucose 6 phosphate. See here the phosphoglucoisomerase enzyme is a work. Then here the phosphatases enzyme is a work, and this glucose inorganic phosphate is there, and this glucose it is increase the osmotic pressure and the stomata become open. So the student in night condition, this glucose plus inorganic phosphate it is converted into glucose 1 phosphate with the help of ATP investment here, and this glucose 1 phosphate it is converted into the starch at the 5 pH. Okay, so the student this is regarding to the mechanism of opening and closing of stomata according to the steward hypothesis. So the student, this steward hypothesis it's also having some objections. See this objection, some of guard cell especially that of monocot plant do not have a starch, so this theory cannot explain the mechanism of opening and closing of the stomata in a monocot plants. So the next objection, it has been observed that the stomata often remain closed at midday time without any change in a starch content. Then the third objection, the theory accounts for utilization of energy for closing while opening does not require any energy. So next objection, in some instance, starch is formed at a pH found during the opening of the stomata. And last objection is it fails to explain the effect of blue light on a stomatal opening. So the starch sugar conversion is too slow while the stomatal movement is a very fast process. So these are some objection to the steward theory. So the student, the next part of our lecture is the active potassium transport mechanism, or it is also called as the active potassium pump theory. It is one and same. Active potassium transport mechanism or the active potassium pump theory. So the concept of potassium ion transport was given by the Fugino in 1967. It was supported and elaborated by the Leavitt in 1974. It appeared to be active mechanism because it's required the ATP in. See, the role of potassium in stomatal opening is now universally accepted, and this was observed for the first time by Fugino in 1967, that opening of stomata occurs due to the influx of potassium ion into the guard cell. Further, the Leavitt and Rasky in 1974 proposed this potassium theory. The opening and closing of stomata is a result of active transport of potassium into guard cell and out of it. The adjacent epidermal cell, it is the subsidiary cells, acts as ion storage for guard cell. So the student, in a subsidiary cell, it is a storehouse of the potassium ion. The uptake of this potassium ion is balanced by uptake of chloride ions. So it has been demonstrated that the stomatal opening induced by the light is accompanied by the transport of these potassium ions into guard cell from the adjacent cell. The organic acid provides the proton or the hydrogen ion in exchange of the potassium for balance. The increased accumulation of ions, the potassium, it's having a positive charge, it is the cations, and organic acids are anions, means negative charge, induce the osmosis of water from neighboring epidermal cells, means subsidiary cells, into the guard cell. The increased turgor pressure results in a stomata get open. So here the explanation of Leavitt concept. In the guard cell, starch, it is converted into the malic acid in presence of light during the daytime. See here, the starch plus water, here the hexose sugars formed, and this hexose sugar plus oxygen, there is a formation of malic acid. Then this malic acid then dissociated into the malate anions, and there is a formation of the proton. See, this is malic acid, it get dissociation, breakdown into the malate ions, and then there is a formation of the protons. So the protons thus formed and used by the guard cell for uptake of the potassium ions, means here the exchange of the protons by potassium, and this is an active ion exchange, required the ATP and the cytokinin, it is a hormone. In this way, the concentration of the potassium ion increases into the guard cell. So at the same time, the cell sap in the guard cell is also increases, and simultaneously the pH become more than 7 and the medium become alkaline. There is also an increase the uptake of the chloride ion. See here, see, this is the vacuole, okay, then this is the guard cell, okay, and here this is the subsidiary cell, outer atmosphere, it is subsidiary cell, then this is the guard cell, and this is the vacuole of the guard cell. Okay, see here, when the, see here, the starch, it is converted into the sugar, okay, and see, there is a, this hexose sugar, it is converted into the malate, okay, and malate get dissociation, it is, see, there is malic acid is a form, and then it is get dissociation, formation of malate and the protons, okay. So the there is also increase the uptake of chloride ion, means anion, by the guard cell to maintain the electric and ionic balance the inside and outside the guard cell. See here, when the potassium ion comes into the guard cell, this potassium ion is also followed by the chloride ions, and it is also come into the guard cell to maintain the ionic equilibrium or electric equilibrium. The malate ions formed into the guard cell are neutralized by these potassium, and thus result into the formation of there is a potassium malate. Okay, there is a formation of potassium malate. Okay, and this potassium malate enter into the cell sap of the guard cell, and this reduce the water potential while increase the osmotic concentration or osmotic pressure of the cell sap of the guard cell, and hence there is a endoosmosis is there. Okay, and result of the endoosmosis, the guard cell become turgid and the stomata it get open. See, it also observe that the CO2 concentration is a low and around the guard cell during the daytime. This is due to the high photosynthetic utilization of the CO2, and it help in opening of the stomata during the daytime. So there is a reverse condition is there, closing of stomata in absence of light, means in a dark condition or night time. So the student, the absence of light, then there is a decrease the concentration of malic acid in the guard cell. Efflux of potassium ion from the guard cell. See here, the from that guard cell, this potassium ion comes into the epidermal cell, while the protons ions come into the guard cell. So here, the efflux of potassium ion from guard cell into the subsidiary cell, while influx of that proton from subsidiary cell to the guard cell. So the loss of the chloride ion from the guard cell increase the CO2 concentration in the guard cell due to the release of CO2 in a respiration. It's combined with the absence of photosynthetic activity in a dark period. The presence of the plant growth hormone here, the abscisic acid, it is also called as ABA. It's inhibit the inhibition, the loss of turgidity, and the loss of kidney shaped by guard cell. So this absence of photosynthesis in a dark condition and presence of the plant growth inhibitor hormone abscisic acid, the loss of turgidity and the loss of the shape of by guard cell, and all these conditions represent the reversal of the daytime event. Under this condition, the guard cell lost the water by hexosmosis and the guard cell become flaccid, and this causes the closing of stomata during the night time or in absence of the light. So the student, see, this is regarding to the opening and closing of the stomata. So it is very simple. Only keep in your mind during the daytime. Okay, during the daytime there is the malate ions and the protons is produced, and see there is a the potassium ions from subsidiary cell, it is a come into the guard cell along with the chloride ion, and here the osmotic pressure is increases, okay, due to the endosmosis. Then during the night time, okay, or in the dark condition, then there is a no formation of that malate and proton. Then here the accumulation of that uh, potassium and chloride in a guard cell. Okay, then the potassium ion concentration is a more in a subsidiary cell, and here the hexosmosis is there, and the guard cell become flaccid and stomata will be closed. So the student, expected question on this topic. First long answer question is explain Stewart hypothesis of opening and closing of stomata. It is a long answer question. Then the next question, discuss the active potassium transport mechanism of opening and closing of stomata. Okay, this type of question is asked on this topic. So the student, in the next lecture we will focus on factors affecting the rate of transpiration, significance of transpiration, anti-transpirant, then excudation, okay, then the difference between guttation and excudation. All these points or subtopic we will cover in our next lecture. Thank you.